We have a pretty universal medical rule, right? Obesity increases your cancer risk. Full stop.
Yeah, absolutely.
I mean, it's written in every textbook. It is preached in every clinic. But if you walk into an oncology ward right now and actually look at patients getting cancer immunotherapy, you will see something that seems, impossible.
Right. The data is wild.
Yeah. The patients who are technically obese, like the ones carrying excess weight, they are frequently surviving longer and responding better to the exact same treatments.
It is completely counterintuitive. I mean, the medical community actually calls it the obesity paradox.
The obesity paradox.
Yeah. For years, it's been this huge elephant in the room for oncologists. Like, why would this metabolic state that, actively drives tumor growth suddenly act like a biological shield the second you introduce an immune-boosting drug?
And that paradox is exactly our mission for this deep dive. We are unpacking a truly groundbreaking study from Nature. It's titled Diet Microbiome Synergy Underlies Obesity-Associated Immunotherapy Efficacy.
Quite a mouthful.
It is, yeah. But our goal for you listening today is to strip away all that academic mystery and figure out the mechanical why behind this paradox. And to do that, we are going to trace the path from the food on a plate to the bacteria in your gut, all the way to the molecular weapons deployed inside a lung tumor. Okay, let's unpack this.
So, to get to the truth here, the researchers first had to fix a pretty major flaw in how we historically study obesity in the lab.
Right.
Usually, if a scientist wants to make a mouse obese, to study its cancer response, they take this highly reductionist approach.
Which means they just feed them garbage, basically.
Kind of. They just feed the mouse a single high-fat chow. It's basically pellets of pure lard, like representing up to 60% of their daily calories.
Which is very convenient for a lab, I'm sure. But nobody in the real world goes to a restaurant and orders a bowl of lard. I mean, we eat complex meals, right? With varying proteins, fibers, sugars.
Exactly. I mean, human obesity is driven by industrialization and, ad libitum food access, eating highly processed foods whenever we want. So to mirror human reality, these researchers didn't just use one diet. They designed 12 highly specific, complex diets for these mice.
Wow.
Yeah, we're talking a Mediterranean diet, a Japanese diet, vegan, ketogenic, an American diet with aspartame, one without.
Super specific.
Right. A classic Western diet, high fat, low fat, and then 3 diets heavily weighted with specific plant fibers. So the psyllium, pectin, and inulin. And they ran this feeding protocol for 15 weeks.
And 15 weeks for a mouse is, that's a significant chunk of its lifespan. It gives the body real time to establish a chronic metabolic state. And they didn't just look at a scale, right?
No, not at all.
They generated this comprehensive metabolic score for every mouse, factoring in fat mass, how well they cleared glucose, and their serum insulin and leptin levels.
Right. And here is where we see the first major physiological shift. The mice that developed high metabolic scores, the ones on the obesogenic diets, they experience a profound baseline alteration in their immune systems.
Okay.
When researchers analyzed their blood and their lung tumors, they found that the obese mice actually had fewer overall T cells.
Wait.
Specifically, their CD4 plus and CD8 plus T cells were totally depleted.
Let me stop you right there because that just doesn't make sense.
Yeah.
If we're talking about immunotherapy, T cells are they're the elite assassins we want doing the killing.
Exactly.
Drugs like immune checkpoint inhibitors, specifically anti-PD-1 therapies, they work by taking the molecular brakes off those exact T cells so they can recognize and destroy cancer.
So if the obese mice have fewer T cells in the tumor environment, how is that an advantage? I mean, you can't release the brakes on a car that isn't there.
That is the crucial tension of the whole study. See, the obese environment is chronically inflamed. That low-grade inflammation, it tends to suppress and exhaust those elite T cells over time.
Oh, I see. And to fill the void, the body just floods the tumor with myeloid cells, specifically patrolling monocytes and neutrophils.
Okay, let's give the listeners an analogy to lock this in. It sounds like a general sending a massive squad of untrained grunts, the neutrophils, into battle while leaving the elite special ops, the T cells, at home.
That's a great way to picture it. The lymphoid to myeloid ratio just plummets. In a normal setting, this is a terrible environment for fighting cancer.
Because the grunts can't get the job done.
Right. The tumors grow aggressively. But here's the twist. The moment the researchers administer the anti-PD-1 immunotherapy drug, the mice with the highly metabolic obese profiles responded spectacularly.
Wow.
Yeah, out of the 6 obesogenic diets, 4 of them, so the American, the aspartame, the Western, and the high-fat diets, caused the tumors to rapidly shrink.
So even with a depleted baseline of T cells, the moment the immunotherapy removed the brakes, those remaining T cells somehow woke up and hit way harder in the obese mice than in the lean mice.
Exactly. They hit incredibly hard.
Okay, but this brings up an immediate contradiction for me. If physical fat is the key to supercharging this therapy, why didn't every diet that caused weight gain work?
And that's exactly where the whole fat equals cure theory falls completely apart. Take the Mediterranean diet, for instance.
Okay.
It caused the mice to gain weight and hit a high metabolic score. But when those mice were given the immunotherapy, it failed completely.
The drug just didn't work.
The tumors completely ignored the drug. Conversely, the inulin diet, which is heavy in a specific plant fiber, kept the mice completely lean, yet their tumors shrank beautifully when treated.
Okay, so let's do a quick recap for you listening to make sure we're all on the same page. Point 1, we have 12 different diets. Point 2, obesity-inducing diets decrease overall T cells, but increase those patrolling grunt cells. And point 3, this paradoxically makes the tumors highly vulnerable to immunotherapy. But the big question for you to chew on is this. If we have fat mice failing treatment and skinny mice succeeding, that definitively uncouples the actual body fat from the clinical outcome. If the fat itself isn't pulling the strings, what is?
What's fascinating here is it's the gut microbiome. The researchers realized the diet wasn't just feeding the mouse. I mean, it was feeding the trillions of bacteria in the mouse's digestive tract.
Right.
To prove this, they used something called 16S rRNA sequencing on the fecal matter.
Let's explain that term for a second. 16S RNA sequencing, think of it as a genetic barcode scanner at a grocery store, right? It just allows you to take a quick census of every bacterial family living in the gut.
Perfect analogy. And what that barcode scanner revealed was a stark divide between the winners and the losers. The diets that responded to the cancer therapy, regardless of how much the mouse weighed, were heavily enriched with a phylum of bacteria called Bacillota, specifically a family called Lactobacilli.
Wait, primarily the genus Lactobacillus?
Yes, exactly.
Lactobacillus. Okay, I know that one. That's like the probiotic poster child.
Yeah.
It's in, yogurt, kimchi, fermented foods.
Exactly. Meanwhile, the non-responder diets, the ones where the tumors just kept growing, they were dominated by a completely different bacterial genus called bacteroids.
Oh, wow.
But, the researchers wanted to prove beyond a shadow of the doubt that the bacteria were in charge, not the slow accumulation of physical fat. So they set up this time-lapse experiment. Instead of waiting 15 weeks for the mice to become obese, they tested them at just 3 weeks.
Oh, that's clever. Because 3 weeks in, the mice haven't accumulated any significant fat mass at all. Metabolically, they're still super lean.
Right. But microbiologically, their guts were fully transformed. The bacterial populations adapt incredibly fast. Yeah. And when they gave these 3-week-old skinny mice the immunotherapy, they responded identically to the 15-week-old obese mice.
That's crazy.
The therapeutic success arrived the exact moment the microbiome stabilized, long before the fat arrived.
Okay, if the microbiome adapts that quickly, how fast can we push it? Because, human digestion takes time. The researchers did a 48-hour diet switch experiment that I think really tests this limit.
Oh, this is one of my favorite parts.
Yeah. They took mice on the psyllium fiber diet, which was one of the worst non-responder diets, totally dominated by bacteroids. And just 2 days before giving them the cancer drug, they swapped their food to the high-fat responder diet.
And the transformation was almost violent in its speed.
Yeah.
I mean, in just 48 hours, the bacteroids plummeted, the lactobacillus bloomed, and most importantly, the previously resistant tumors suddenly shrank when hit with the anti-PD-1 therapy.
Okay, let me challenge the mechanics of that. 48 hours is, it's barely enough time for a mouse to fully digest and pass a few meals.
Yeah.
How is it physically possible for an entire biological ecosystem to restructure itself and, alter an immune response in the lungs in 2 days?
It really comes down to the replication speed of gut bacteria. A lot of these microbes can double their population every 20 to 30 minutes if they have the right fuel.
So it's not like slowly remodeling a house over months. It's more like a manufacturing plant. Let's use an image memory here. Imagine a factory. The factory machinery, the bacteria, is always running. But if you change the raw materials arriving at the loading dock, the output changes instantly.
That's a brilliant way to conceptualize it.
Right. If you stop delivering steel and start delivering rubber, the factory immediately stops making car frames and starts churning out tires.
Exactly. By switching from psyllium to the high-fat diet, you instantly changed the raw nutrient materials. The lactobacillus machinery went into overdrive. And when researchers looked inside the tumors of those 48-hour switch mice, The T cells weren't just present. They were hyperfunctional.
What were they doing?
They were pumping out massive amounts of interferon gamma and tumor necrosis factor. These are the highly toxic chemical proteins that T cells use to initiate tumor cell death.
But, and this is key, if they flipped the experiment, like taking mice established on the high-fat diet and switching them to psyllium 48 hours before treatment, the therapy was totally ruined. The factory lost its premium raw materials, the production of those immune-boosting chemicals stopped, and the T cells just went quiet.
Precisely. Which sets up the ultimate biological showdown, really.
Okay, what showdown?
Well, if the diet dictates the microbiome so powerfully, what happens if you try to force the wrong bacteria into the right diet environment? Like, which one exerts more control over the immune system, the food or the bug?
Here's where it gets really interesting. This is where we get into fecal microbiota transplants, or FMTs. Basically, taking the microbial ecosystem from one subject's poop and physically implanting it into another.
Yep. So the researchers took mice eating the high-fat diet, which is our proven responder fuel, but they gave them continuous fecal transplants from mice on the psyllium diet, the non-responders.
So they are constantly seeding the gut with bacteria that are known to fail against cancer. Let's go back to our factory analogy. You're importing terrible factory machinery, but you're still providing premium raw materials. Who wins?
The raw materials win, every time. The diet overrode the transplant.
Wow.
Yeah, despite constantly receiving the bad non-responder bacteria, the mice on the high-fat diet still responded beautifully to the cancer drug. The high-fat fuel was so powerful that it essentially forced the transplanted microbiome to completely reshape itself into a responding profile.
Okay, let's pause for another quick recap. Point 1, the diet-gut axis dictates therapy success, not body weight. Point 2, Lactobacillus is our hero machinery. And point 3, changing the diet can alter tumor sensitivity in just 2 days. It can even override a bad microbiome transplant.
Exactly.
But, mice are one thing. The question for the listener is, does this hold up with human biology? If we take human bacteria, does the diet still win?
It does. They actually scaled this up using human stool from a lung cancer patient who had completely failed anti-PD-1 immunotherapy. This was a confirmed clinical non-responder.
Okay.
They transplanted this failed human microbiome into 2 groups of germ-free mice. These are mice that have absolutely zero bacteria of their own. One group was fed the psyllium diet, the other the high-fat diet.
And the results mirrored the mouse-to-mouse experiment perfectly, didn't they?
To a T.
The mice on the psyllium diet, burdened with the failed human microbiome, saw their tumors grow. Treatment failed. But the mice on the high-fat diet, carrying the exact same failed human bacteria, successfully fought off the tumors. The nutritional environment completely overrode the human donor's clinical failure.
And at this stage, the researchers needed to identify the exact microbial machinery driving this victory. So they used something called advanced shotgun metagenomic sequencing.
Shotgun sequencing. How is that different from the barcode scanner we talked about earlier?
Well, the 16S barcode scanner just tells you what families are present, like a general roll call. Shotgun sequencing looks at all the genetic material in the sample, allowing researchers to see exactly which specific species are thriving and exactly what functional genes they carry.
So sticking with our analogy, they aren't just looking at the factory building anymore. They're looking for the specific worker inside the factory doing the job.
Exactly. And they found a major synergistic hero, a specific species called Lactobacillus johnsonii.
L. johnsonii. Okay, if L. johnsonii is the elite worker, the obvious clinical question is, why don't we just manufacture a probiotic pill packed with L. johnsonii, give it to cancer patients, and solve the problem? Why deal with complex diets at all?
Because, as we saw, the worker is totally useless without the right raw materials. They proved this by taking a new batch of germ-free mice and monocolonizing them. They introduced only L. johnsonii into the gut, absolutely no other bacteria.
Right.
When they fed these L. johnsonii mice the psyllium diet, they only achieved a partial, weak response to the immunotherapy. The tumors didn't clear.
So the probiotic alone isn't a magic bullet.
Not at all. And to check the other side of the equation, they took mice, gave them the high-fat diet, but colonized them with a completely different bacteria, Myribaculum gordon-carteri. Again, only a weak partial response. But when they combined the specific worker, L. johnsonii, with the specific raw materials of the high-fat diet, they achieved near-total tumor clearance.
It demands synergy. I mean, you can have the best factory machinery in the world, but if you feed it the wrong raw materials, it can't build the weapon. You can have the best raw materials, but without the right machinery, they just sit on the loading dock. You absolutely need both to manufacture the cure.
That's exactly right. The biological synergy is non-negotiable.
Okay, let's do our third mini recap. Diet is the ultimate boss. It overrides bad transplants.
Yeah.
And the ultimate anti-tumor weapon isn't just a food or just a bug. It's the precise combination of an obesogenic diet and L. johnsonii.
Perfectly said.
But this leaves us with a glaring geographical problem. The factory, right, the gut microbiome processing the high-fat diet, is located in the colon. The battlefield, the tumor being attacked by the T cells, is located all the way up in the lungs. Bacteria don't exactly swim up the bloodstream to hand-deliver weapons to T cells. How is the gut actually communicating with the lungs?
Right. To bridge that physical gap, the researchers turned to untargeted metabolomics. Instead of looking at the bacteria themselves, they scanned the blood serum of the mice to catalog thousands of tiny molecules, or metabolites.
Metabolites.
Yeah. These are the chemical byproducts created when bacteria digest food. They were looking for the molecular messengers traveling from the gut to the lungs.
The actual weapons being shipped out of the factory.
Precisely. And the data pointed brightly at aromatic amino acids, specifically the metabolic breakdown of an amino acid called tyrosine. When L. johnsonii breaks down the tyrosine found abundantly in the high-fat diet, it produces a specific phenylpropionate metabolite called DAT, disaminotyrosine.
Okay, I want to give the listener a memory trick here for DAT. Let's call it the direct attack transmitter, DAT, because it's transmitting that attack signal directly from the gut to the lungs. So DAT is the cargo. When they checked the blood of those germ-free mice that had both L. johnsonii and the high-fat diet, what did they see?
Massive, highly concentrated spikes of serum D8. That specific bug and that specific diet essentially formed a biological DAT factory, just flooding the bloodstream with this molecule.
But what is the direct attack transmitter actually doing when it reaches the lungs? Because to figure this out, the researchers bypassed the gut entirely, right? They took T cells straight out of a tumor, put them in a petri dish, and bathed them directly in DAT.
And the reaction was explosive. DAT doesn't just bump into a T cell, it acts as a potent molecular signal that alters the T cell's internal programming. When exposed to DAT, these T cells became hyperactivated. They radically upregulated their production of granzyme B.
And granzyme B is, it's essentially the toxic payload. It's the enzyme T cells inject into cancer cells to trigger apoptosis, forcing the cancer cell to self-destruct.
Yes, the DAT essentially arms the T cell to the teeth. So when the anti-PD-1 drug removes the regulatory brakes, the T cells aren't just waking up. They're waking up heavily armed and hyper-aggressive, all thanks to the DAT supplied by the gut.
It's a beautifully clear mechanism. But as always with mouse studies, the ultimate question for you listening is, does this deeply specific pathway actually dictate survival in human beings?
Well, the researchers took this exact molecular pathway back to the clinic. They analyzed blood plasma samples from a cohort of 53 human patients with non-small cell lung cancer who were actively undergoing immune checkpoint inhibitor therapy.
This is where the rubber meets the road. Did the human responders have the same chemical messengers?
They did. The patients who successfully responded to the therapy had significantly elevated blood levels of a molecule called 3-HPP sulfate.
And 3-HPP sulfate is directly related to our DAT factory.
It's a direct downstream product. When DAT enters the human liver, it gets processed into 3-HPP sulfate. So finding high levels of 3-HPP sulfate in human blood is the definitive fingerprint that this exact tyrosine processing microbial pathway is highly active in those patients. They also found high levels of ILA, which is a similar messenger derived from the breakdown of tryptophan.
Wow. And to tie it all the way back to our very first paradox, the obesity paradox, they performed one final, brilliant human-to-mouse fecal transplant. They took stool from human donors, but this time they separated the donor strictly by their body mass index.
Right. They transplanted microbiomes from low BMI donors, so under 25,
Yeah.
considered normal or lean weight, and high BMI donors, 25 or over, considered overweight or obese.
And what happened when those mice were given the cancer drugs?
The microbiomes from the lean human donors failed to sensitize the mice to the immunotherapy. The tumors grew. But the stool from the high BMI human donors successfully sensitized the mice. The tumors shrank. It proves that humans with a higher body mass index naturally harbor a more robust, primed microbial ecosystem, one that is highly capable of producing these vital chemical messengers when given the right fuel.
Okay, final recap for you. Point 1, bacteria eat the fat, produce metabolites like DAT, our direct attack transmitter, which enter the bloodstream. Point 2, natia acts like a chemical steroid for cancer-killing T cells. And point 3, high BMI humans naturally harbor more of this specific microbial ecosystem.
Exactly.
So what does this all mean? Bringing this all together, the obesity paradoxes, well, it's a bit of a misnomer. It isn't that carrying excess adipose tissue is intrinsically protective against cancer. It's that the diet required to become obese accidentally cultivates a specific, heavily armed microbial ecosystem.
That's the key.
The physical fat is just a visible byproduct. The microbiome and the metabolites it produces are the actual engine driving the immunotherapy success.
Exactly. And the clinical application of this is incredibly exciting because, I mean, no oncologist is ever going to advise a cancer patient to consume a high-fat, highly processed diet for years just to build this microbiome.
Right. The systemic risks of long-term obesity, like cardiovascular disease, diabetes, and even generating new cancers, they far outweigh the benefit.
But because we know the microbiome can pivot in just 48 hours, we don't need years.
We just need days.
Precisely. This research paves the way for short-term, acute interventions. We can use highly targeted dietary changes, perhaps just a few days or weeks of a specific nutritional profile, combined with targeted probiotics like johnsonii or even direct metabolite supplements like date, to temporarily hack the patient's immune environment right before we administer the cancer drugs. We prime the factory, manufacture the weapons, and then remove the brakes.
We've spent so much of medical history obsessing over how to attack the tumor directly, looking at the scan, trying to cut the cancer out, or poison it with chemotherapy. But what this deep dive reveals is a profound paradigm shift.
It really is.
The future of oncology isn't just about the chemicals in the IV bag. It's about the microscopic machinery in the gut and the specific raw materials we feed it. So I want to leave you with a final thought to mull over. Next time you sit down for a meal, consider the biological factor you're operating. Who are you really feeding? And what kind of cellular weapons are they building for you?